Perfect. Okay. So let me go to the presentation. View full screen. Okay. So I'm going to talk a little bit about sequence mapping. This sequence mapping lesson can be applied not only for RNA-seq, but for anything. But RNA-seq has some specific complexities. I'm going to be technical in some points, but then don't worry because I will tell you, by the way, you can forget about this, okay? But still it's good to know to have these things in a slide if one day you need it. So the outline of topics of these lessons is going to be more these 5. The 6 is is one slide that, because this comes from a different class. So will be basics of read mapping, short read mapping, along with mapping of transcriptomic reads, that is the important one for you, and then evaluation and visualization of mapping reads. So let's start with basic of read mapping. So what is a read mapping? And this is an important concept. A read mapping is the process of search the location, okay, first is the location of a read comparing its sequence with the sequence of a reference, okay? So basically, the read mapping is this thing that you can see here. This is not a read mapping. This is the read mapping, telling me that my read map in the position 10 to 23 in this reference. So what is this then? That is the thing that is called a sequence alignment. And it's not necessary to have a sequence alignment to have a sequence mapping, okay? You may produce a sequence alignment if you want, but this is not completely necessary. Okay, a sequence alignment is a way to arrange the sequence of DNA, RNA, or protein to identify regions of similarity that may be consequential, functional, structural, or by evolutionary relationship between sequences. Aligning sequence of nucleotide or amino acids are typically represented as a row in a matrix, but as it's not a location, it's a row in a matrix. When you are doing sequence mapping or read mapping, sometimes you have both, okay? You have the sequence alignment and the read mapping. But the thing that is important for us is the read mapping. We are not interested in the sequence alignment, at least not for this RNA-seq. If you are looking, for example, for SNPs, structural variation, and so on, that is essential. But for the read mapping, it's not, at least not at this point, okay? So that is important difference. When you are doing a read mapping, any read mapping, you have to have in mind several considerations. One of them is the read of the length. The other is number of reads, size of the reference, and differences with the reference. All these things are going to impact the mapping. So in some ways, the read mapping will be a process that you have a reference, you have a lot of reads, and then during this process, you start to align them, or may align them or not, but at the end, you are going to reveal the positions of them. Not like you can see this example. This schema, no, here, that will be the mapping. When we're thinking about the read length, you have a really wide range. Of course, you can map next generation sequencing short reads. That is the one that we are doing mapping now. That the sequence length goes usually between 25 and 300 base pairs. Then you can have Sandia reads or long fragments from PacBio. You may sequence or may want to align genes transcript fragments, so you are going to increase the sizes. Next generation sequencing long reads will be here, a little bit between these 2 points. You even can align, if you want, contigs or scaffolds or even chromosomes between them. No? So you have this wide range and usually go from 25 and 300 base pair to 1 megabase. And of course, the programs that you use to do this are going to be different. So the programs to align this are going to be different from the programs that you use to align this. These are going to be less time consuming. This will be more. Okay. Other thing that you need to keep in mind when you have aligning reads, mapping reads, is that shorter that is the reads, more possibilities of ambiguity do you have. So let's suppose that you have this reference, okay? And you have these reads that you can see here with 1, 2, 3, 4, 5, 6, 7, and 8 nucleotides. If I'm trying to map these reads in this reference, potentially I can map in 2 positions, this one and this one. But if I add one extra nucleotide and instead 8 I'm mapping 9, you can see that in this case you have a differential nucleotide here, so this read would map only to this position but not this one. So the length of the read is a critical factor to align read specifically. So longer that is the read, more specific that should be the alignment. The number of reads also is an important factor. Usually, when we are aligning short reads, we can align from the order of millions, and then when we move up or well down in this graph, we will be reducing the number of reads. So usually we map dozens of chromosomes. We don't map billions of chromosomes. Okay, that in some ways keep this balance between computational resources used in a good way. No, so genes transcript usually have 10,000, 100,000. Okay. Context scaffold, the same thing, and then for chromosomes you will have only dozens of them. So these two factors are going to tell you about the computational resources. So more reads that you have and longer that is going to be the reads, more computational resources that you will need. Of course, you have some regions here that are prohibitive. So of course, mapping millions of chromosomes will be prohibitive. Mapping thousands of chromosomes still could be tough for the computer. Okay, but mapping, for example, thousands of genes or medium-sized reads is doable, and of course dozens. Of course, mapping dozens of short reads will be something really extremely fast and effective. No, so that will this graph more or less tell you about the resources that you need. The size of the reference also is an important consideration, no? Because the smaller that is the reference, faster that will be the search. It's not the same to look for this pattern here than here. And also differences with the reference will produce, will speed up or not the search. No? When you have mismatches that you can see here, probably that will take more time. No? You can have mismatches like this one, or you can have gaps either in the reference or either in the gap in the reads. No, so that also will slow down these searches. More difference with the reference, more difficult that will be to align the map. No, to align and map the read. No, so here will be easier, but as soon as you have more and more differences, will be more difficult to align. No, this one will be much more difficult than this one. Okay. So that is another important factor. One important thing that we can define when we are mapping reads is the mapping sensitivity. The mapping sensitivity is the ratio between reads mapped correctly with reads mapped incorrectly. And of course, we only can evaluate how many reads we have mapped incorrectly if we really are creating a model. Okay, so for example, we take a genome and we break this genome in small pieces. We know where these reads should be, and if we map them back, we will be able to estimate mapping sensitivity, for example. Okay. Other thing that's important is higher that is the sensitivity and higher that is the speed of the mapping, more computational resources that you are going to need. If your sensitivity and your speed is low, you are not going to need more. Okay. A lot of computational resources, but more than you move in this graph, more computational resources that you will need. Okay, that is the important part. That is the part that I would like you to keep in mind. Then I'm going to tell you what are the different mapping methodologies, but this is something that sometimes is kind of complex, so don't worry. Okay, that is kind of, I want to give you just a general vision, but you don't need to keep that in your hard drive, okay? Your mental hard drive if you don't want it. There are different types of mapping methodology. You have hashing methods and suffix array methods, okay? A hashing method, the thing that you do is you have a reference, you have a read, you decompose the read in small fragments called k-mers, and once you have the k-mers, you look into your reference for a perfect match. Once you have the perfect match, you do the extension. So this process is important because you are looking for the thing that is called a perfect seed hit. Smaller, that is going to be the seed, faster, that will be the process. But also keep in mind that, as we mentioned here, more gaps that you have, more difficult. So if you have too many gaps, maybe you are not going to be able to find the perfect seed hit. So that is a problem. So when you are using mappers, sometimes it's important to know what is going to be this seed, okay? Let's put in this way. If you are mapping Brassica rapa to Arabidopsis, by default, you will have a low number of reads. But then if you want to, maybe you can play with this seed length, doing this seed length smaller, and then you will increase the number of reads that map. At the same time, you may have some false positives too. Once the hits is found, you do have a stage of extension, and you have different mechanisms, different algorithms to do the extension. No, you have the Needlen-Gush or the Smith-Waterman, that is the most common one. Small is the K, more sensitive but slower. High K, more specific and faster, but bigger is going to be the databases for search. Okay, so that is the two observations that you should know here. Of course, you are not going to do this exercise. So just this is an explanation about how these indexes are created in the reference to look for these k-mers. So basically, the thing that you do is this Borrow-Weller transformation. Okay, so you are going to do these indexes when you are going to run the high seed program. Okay, so the first thing that you need to do is you need to create an index of your reference. An index of your reference, or these indexes of the reference, because it will create more than one file, is a decomposition of your reference in a way that is going to be much, much, much faster for this program to look for these hits. Okay, so basically, the thing that you do is you have an input. Let's say that you have this sequence that is banana. Then you will do a transformation, doing rotation. So you will transform this banana in different words. Okay, so you will have banana, but then and then the end, and then you will have the beginning and banana, and then you will move one until you have all the different possibilities. Okay, this represents the beginning of the string, and this represents the end of the strings, and then you will have all of these different possibilities. When you have that, you sort them lexically. So first A, B, N, beginning of the line, end of the line. You take the last column. Okay, so this is the lexicon order. Then you take the last column. So this is the last column, and then you get this output. And this output is going to help a lot, okay, to search very efficiently for these patterns. Okay, so that is hashing methods. I'm not going to go into much detail, just telling you that you need to create an index, and the index is done in that way, and this is a more efficient way to create an index. Then you have these suffix array methods that are based in suffix array lists. Okay, so basically the thing that you do is you take a reference and then you build a suffix tree. No, so this is a reference that you have with 10 nucleotides, and this will be one possibility. But then you take how many of them can you build based in A. So if you start with A, so you are looking for A. This is the thing that you can look. But then if you look for T, you have this T here. So you can build this branch in this tree, or this T here, that will be this different branch. Then you go to the next one, that will be a G. And for the G, you have this branch. Then you can have the second branch, but because the second branch overlap with the first one, is there, it's the same branch. And then this one, this GGC or GC still overlap with this branch, so it will not be a different branch. Or, and then you go after the G, you can have also the branch of Cs, the branch of Ts, or if you go to the next one, that will be the C, that will be your last branch. Okay, And then when you have a new read, you go to your suffix tree and say, okay, so my read is G. So I will go to my suffix tree. This is G. The next one is C, C, G, G, T, T, G. Okay, and it's how you will find the reads, the map. Okay. This is very fast, but when you have errors or when you have polymorphisms, the search space that you are need to look in these trees are very, very high. No, so that will be all. Okay, so that is the methodology. As I said, keep in mind, the only thing that you need to keep in mind is that you have two methodologies, suffix array and hashes method, but you don't need to go into these details. I think that, yeah. Questions that you may have of this first part?
Just a tiny question. Can you use both methods together? Can you combine them, or do you normally use just one of the two?
So usually the tools use only one of them, but now that, I mean, there are a lot of different tools. So some tools tend to do some kind of a hybrid method. The thing is, you cannot find tools that use only suffix array, because suffix array is a mathematical operation that is very efficient and is great, sounds great, but when you try to escalate this with a lot of reads, it's inefficient, it's too much. So this was, so all generation mappers use suffix array, but as I said, that was when you needed to map just 2, 3, 4 sequences. Once that, now that we need to map thousands, billions of them, suffix array methods are impractical. But still, some programs tend to use this suffix array for some steps. So yeah.
Okay, thank you.
Yeah. Shortrange mapping. So I will tell you about different mapping software. Okay, and I will tell you also one of the important things is that when you are producing the mapping, you may have gaps or not. So you may have indels or not. Okay. So the most common short-read mapping software, they don't produce gaps. This is the most popular one. So you have I2TA2020, you have BWA, GEM is another one, Novaline, SOP, or Stampy. The most popular ones for sure are BWA and I2TA20. So a good example of using I2TA will be something like this. No, you have your program I2TA. You can have some options that you will, I mean, if you print the help, you will find them. Then you have your index that you have previously created. Then you have the first pair of your reads, your second pair. Maybe you have the reads intervaled, so that will be the option. That is not common. Or you have single reads, and then you will have the outputs. No? So that is how usually these commands works. Once you run the program, the output that you will have is this SAMBAM file. And just remember that SAMBAM files, as I mentioned in the last Tuesday, is a file, text file, if SAM, or binary file is BAM, where you contains a header. So the header is usually all the lines of the header start with @ so @HD, @SQ, and so on. And then once the header, you have the second part that is going to be 11 columns. Each of the rows will be one of the reads that you have mapped. And then you have, for these 11 columns, they have different meanings. No? So the first column tells you about your query name or the name of the read. The second is a flag that tells you several parts of the information if the read has been mapped, it's not. The reference, sequence ID, so in this case it's ref, but could be chromosome 1, chromosome 2. Then you have a map queue that defines how good or bad was the mapping. Then you have the cigar line that tells you about the alignment, how the alignment is. You have matches, insertion, deletions, soft scripting, and so on. Then you have rnext and pnext. That means for the next, if you are working with pairs, it's telling you if the pairs has been mapped or not, and also the quality of the mapping. Then the length of the mapping, and then the sequence and the quality. The one that is more important in the some files are the flag, as I mentioned last time, and the flags can tell you things like if the segment has been unmapped or the pair has been unmapped, or the sequence is mapping in the reverse complementary, or the pair is mapping in the reverse complementary. If it's the first read of the map or the second read, you have secondary alignments, or sometimes you can mark things like not passing quality controls or PCR duplicates. So for example, if you have a read that you have the flag 4, means that the 0x4 flag is activated, and that means that the read has been unmapped. You have 16, for example. 16 means 0x10, so means that the read reverse is a read reverse 10. Or you have 83, means that it's a combination of the read is paired, the read has a proper pair, and then the read is a reverse stem and is the first in the pair. If you are more curious about this, because this is kind of a complex way to set up flags, you can go to this atlas and they will tell you, no, that's that. Okay. I think someone trying to request control for the presentation. Please don't do it. Okay. When you need to manipulate some BAM files, this is essential. The tool that you are going to use so far is going to be some BAM, the SAM tools. Okay, SAM tools is a complex. SAM tools is a complex a group of tools that you can use to do complex operations, from change from SAM to BAM formats to sort them. Sort the reads, because when you are mapping, the output is going to have the same order than the input that you was reading. So the first read in the output will be the first read in the FASTQ file. The second read in the output will be the second read in the FASTQ file, and so on. But that doesn't mean that in the chromosomes the first is at the beginning and the second is after. Maybe the first is going to be at the end of that chromosome, and then the second is going to be at the beginning of the chromosome. No, so you want to sort by reference coordinates. You need to use this SAM tool sort, and you will use indeed. You can index files, or even you can do, as I said, a lot of different operations. So that will be for short read mapping. Let me go to long read mapping, and then I will stop and ask you for questions. Long read mapping, you have two different types of tools. You have traditional tools adapted for genomic comparisons. And for that, of course, there are many, but the ones that I use more and I recommend are MOV, LASZ, BLAST, and BLAT. MOV and LASZ are adapted for long sequences. You can compare, for example, chromosomes if you want. It can take a while, but you can compare chromosomes. The output sometimes is kind of a different complex output. So MOV is fast, but the output is a backbone and XMFA file with alignments that sometimes is difficult to work with. LASZ can have math, or the most common one that is SAM, so that is more convenient, but this is kind of a very slow program. For medium sequences, you know BLAST already, and you also know the BLAST formats, maybe. And then BLAT is kind of a new version of BLAST, or a different version of BLAST, that also is kind of a fast and is commonly used to map genes, for example, or transcripts. When you are thinking about long read mapping, one of the difficulties, remember, is that the long reads have a lot of errors. So it's the thing that we call noisy. So you need to perform a noisy mapping. So it means that you have a lot of the thing that you are going to compare are can have errors. No? So this is the thing that is wanted to represent here with these red stars. No? That you may have a lot of errors. And to do that, of course, the program that I showed you before may not work. So, and you want to do it faster, because MOV, for example, is not aligning a lot of sequences. It might be maybe 5 chromosomes against another 5 chromosomes. But when you have a lot of long reads, you may have 100,000 or even millions of long reads. So you have this long read mappers that are noisy mappers. So they deal with these problems about having errors with the sequences. BLAST-R was the first one, and GMAP was the first generation of these mappers. That BLAST-R works fine. GMAP is terrible from my point of view. And then you have a second generation of mappers that you have, for example, Minimap2, NGMLR, and LORFAS. And these are pretty good. I really like Minimap2. No? So when I need to map reads, I usually use Minimap2. So that is another generation, just to keep in mind. All of them have the output of SUM, and you can have the input of FASTA or FASTQ. Okay, questions about short or long read maps. I'm talking about any kind of mapping, okay? They usually you use these mappings when you are mapping DNA, okay, or things that come from the DNA to another DNA. So it means genes to genomes, or yeah, these things. No? You resequence a genome and you want to map against another genome. This is the kind of tools that you use. Questions that you may have about this part.
Sorry, is it the same for microRNAs?
Is the same for microRNAs? No, for microRNAs, you need to map using transcriptomic reads. Oh, well, you can map. I mean, you can map, for example, using bowtie. But the thing is that when you are mapping things that come from a transcriptome, you need to have a Gapid aligner. And the reason is that transcriptome, and as I'm going to explain now, you have intron exon boundaries. And because you have intron exon boundaries, sometimes if you want to, the non-Gapid aligners like bowtie doesn't deal very well with that. So you have a read that map into different places. A fragment map in one place, the other fragment map in another place, and then you have a gap in the middle. This program doesn't deal with that. A microRNA comes from the processing of, for example, an antisense mRNA. So it may, sometimes when you map them, it's not common because they are very small, 21, 23 nucleotides, but it may be in this boundary, so that could be a problem. So, but yeah, by default, I will say that, yeah, you can use bowtie or BWA. No, it's reasonable to use those.
Thank you.
Yeah. Still, there are some specific protocols to map microRNAs. So, yeah. I will say that, for example, so the thing is, few years ago, if you wanted to analyze microRNA, you use bowtie. You didn't care to use top hat, for example, that is a Gapid aligner, because maybe you were introduced to much noise. But now that you have better aligners like HiSat that can deal with everything if you want, you use HiSat. So, okay. So I will talk about that. So when you are aligning transcripts or transcript-derived reads, as I said, microRNA will be, non-coding RNA will be another one. The problem that you have is that when you have your reference genomes, you have introns and exons. So you have reads that can map fully in one of the exons, but sometimes you have reads that you map one part in one exon and you map another part in another exon, and then you will have a gap in the middle. So that is the thing that is called an intron exon boundary. So you have reads that can cross the boundaries. Okay, and these reads are very important because are going to tell you have alternative splicing. No, so you have one read that is come from exon 1 to 2, but then you have another read that comes exon 1 to 3. You know that you have two alternative splicing forms. I will talk about that later. But this is one of the main differences when you are mapping reads with transcriptome, that you have this gap. Okay. So this is something that needs to be taken into account. Well, this should be exon 1 to 3. Okay. So there are a lot of different Gapid align tools for exon intron boundaries. Okay. The most popular ones are TopHat2, HiSat2, and STAR. we will be using HiSat2 and start. When you need to decide which one do you want to use, sometimes you have two options. You can use the most popular one, so you go and see what is the one that people use and use it, and still is fine because maybe you don't need to justify anything with your reviewers. Or you go to a publication and check which one should be the best according to the simulation that they did or the analysis that they did. So this is an analysis, a simulation that they did with different RNA-seq aligners in 2017 using default parameters or trying to optimize the parameters, no? D and T. You have things like CLC. Personally, I don't like CLC because it's a program that you need to pay. You have a graphical interface, so you can forget to some of the processes that you need to do for interact with a Linux server. But then the license of this program is something like 10,000 euros per year. And I think that is excessive. So you have mappers like this one, and then you have open source ones. So for example, Top Hat is a very common use program, but as you can see, it doesn't align correctly a lot of reads. No, it's a very popular one. And then trying to turn them, you may have a lot of misaligns. NoAlign is one that you need to pay for, and it produces good alignments with Gapid and non-Gapid, so you have the two versions. But the ones that we use more is HiSat and Start. And this graph gives you a very interesting, HiSat2 indeed, gives you a very interesting message that is HiSat, that is a very, very, very popular one. It's not a bad tool. Indeed, this tool, when you turn the tool, is better than CLC. But when you use with the default parameters, you may not have a lot of reads aligned correctly. You may not have a lot of reads aligned incorrectly according to this simulation, okay? This is a simulation. But yeah, but then Start, for example, align a lot of reads correctly, and then when you tune, you may increase a little bit, but not. So it means that if you don't want to think much about alignments and alignments parameters, maybe Start is a good option. No? That is the background message here, no? That you need to think which one do you want to use. So that is an important thing. During the exercise, you will run two. You will run HiSat2, and you will run Start, and you will compare. You are not going to run for all the reads. So my recommendation during the exercise is just run with one of them or two pairs if you want, and then compare the results, and then you can decide which one you are going to do the rest of the reads, okay? As I mentioned before, it's good to look for this protocol when they use HiSat, SlimTie, and Valgo for the analysis. Questions that you may have about mapping transcriptomic reads. Okay, maybe these questions will pop up during the exercise. The last part, that, of course, is after that you map, you need to control, say, okay, how good or bad was my mapping? And you can visualize them if you want to. So there are different parameters that you can use to evaluate the quality of the mapping, okay? You can use coverage, so you have the genome, how many, looking for region, how many reads are in that region, is the thing that we call coverage. 1x means that you have only one read, 5x means that you have 5 reads to a specific position, and so on. Distribution of mapping qualities, so some of the mapping qualities are going to be good, some of them are going to be bad, so you can go and look for the distribution. Percentage of read mapped, and so on. When you are running some programs like HiSat or Bowtie, or even Start, you have an output like this that tells you what is the alignment rate. No? So this is the output that you obtain for the first pair of our datasets. And our first pair of datasets, our dataset map 96.75%. So that is a pretty good mapping. The interesting thing is that when you look for this, for example, is you are looking for pairs, okay? So some pairs doesn't align concordantly, or doesn't make sense. Some pairs map more than one time, some pairs map only one time, so you will still have all these different parts. But the one that is more important for you is just the overall. When you are more looking for mapping percentages between 70% and 100% are acceptable. Well, of course, 100% is perfect, but 70% will be okay. When you have lower than 17%, you need to start to look for why. Okay, if you have 75, you can move forward and assume that 25% of the read doesn't map because maybe they are different, maybe you don't have a good quality, maybe you have some contaminations and so on. So, and you still can play with that if you want. Okay, you can just ignore them, say 75, yeah, fine, let's move forward. When it below 70, you need to stop and say why I have so much low mapping percentage. Okay. For the tools, there are tools that you can use to collect simple stats. So common ones are SumTools. So for example, you use SumTools view-c my file is going to return the number of sequences. Or you can use SumTools stats, and it's going to produce a very complex file with a lot of headers that you can look for a lot of different stats. Okay. Before you run these programs, it's convenient to sort the BAM files. Okay, don't forget to sort the BAM files before you run these programs. Okay, so that is some tools. I like more BAM tool stats, and you will use that because have kind of a much better friendly output like this one. No, so you are mapping this total number of reads, and 100% of them has been mapped because I already filtered those. Okay, so this is looking into the filtering, the filter output. So that would be another option. Or Picard tools. Picard tools is good too because also it's going to tell you if you have when you are looking for pairs, it's going to tell you also what is the average distance between pairs. The outputs can be more difficult because this is, if you copy and paste this in an Excel, will work fine, okay? But otherwise, when you look into the screen, it's kind of difficult to see. So typically it works in this way. Then you have packages that can produce more complex outputs, no? The ones that are more popular one are SeqQC and QualiMap. QualiMap, as you can see here, that produce a lot of different things. We are not going to use those, but of course you can use it if you want. I installed QualiMap in the server and produce this summary in an HTML format like FastQC. And then you can see some graphs saying, okay, so just looking into the average, so most of the reads map in the exons, some of them in introns, some of them are intergenic, and so on. So it's pretty good, and you have a lot of different graphs. So it's pretty good to look one by one and evaluate the quality. For me, as I said, we will look into this one for now. That should be enough. Okay, so that is in terms of quality evaluation. Of course, maybe you want to look into the alignments. And there are a lot of different tools. So not all of them has the same functionality. Some of them may change. Some tools are fast. Some of them are slow. Some of them are more intuitive. Some of them not. Some of them can upload also some GFF files. So there are some ones that are good, but maybe you cannot upload annotations. So that could be a problem, and so on. So the most popular tools are BAM Viewer, BAM View, Tablet, IGV, and UG. Personally, I like Tablet and IGV, and are the ones that I'm putting as an example. Tablet is fast, has a nice graphical interview, but the problem is that you cannot upload if you want your annotation. So you cannot upload, for example, a GFF file with your word that telling you what are the genes. You can upload the reference, and then you can upload the BAM files with the read that maps, and you can see the alignment. But you don't know if they are aligning a gene or outside the gene. But still it's fast, so sometimes it's good to work with. And then you have the IGV, or the Integrative Genomic Viewer. It's more difficult to install. Sometimes it's also more difficult to play with. But the advantage is that you can upload the BAM files, but also you can upload annotations. You can upload BCF files or GFF, and then you can check for more complex things if you want. Usage analysis, as I mentioned before, is just one slide that tells you, of course, all these mappings can be used for anything. So you can use for comparative genomics, transcriptomic analysis, epigenetic analysis. So you are doing a chipsec, you will need to use some of these tools too. Okay, not specifically for a mapping, transcriptomes, but some of them like bota is something that you can use too. Okay, questions that you may have about this part or any part of this class lecture?
Yes, I have 2 questions actually. So the first one is, previously you showed the graph where you have all this software and the tuned one and the untuned one, right?
Yeah, that is
and the question is, I mean, how can we know, how can we tune the software in a right way?
That is a really good question. The only way that you can tune, so you have some parameters and some not others, no? So one of them is experimental parameters. As you can see, when you are tuning, most of the time the thing that you do is you increase the number of aligned reads, no? The problem is, so the thing is you don't know if you are aligned correctly or incorrectly, but you can increase the number of aligned reads. So good way to tune them is, so for example, you imagine that you are mapping Arabidopsis to Brassica to Arabidopsis, or Arabidopsis lirata, that is a different species of the Arabidopsis genus, to Arabidopsis thaliana. You are mapping them and you get 80% of the reads. Then you may think, well, because I have some differences, maybe if I reduce the seed size, I will be able to increase the number of mapping reads.
Okay.
So that will be kind of the experimental way to do it. I can increase sensitivity in order to see if I can have more. So that will be a good way to do it experimentally. And then you check if you have more mapped reads. So that would be an easy way to tune them. Just you change parameters and you see you increase the number of reads that you are mapping. Again, imagine that you run the first map and you get 80%, and you say, I can live with 80%, or I would like to scratch a little. Then you can start to change parameters and see if from 80 you move to 85. That would be one option.
Okay. And also the second question is, I mean, let's say I have 30% of unmapped reads. And how can I search for contamination? Because I mean, contamination can be from humans, but also bacteria, fungi.
Yeah.
So how can I search what are those 30% of my reads?
So usually the thing that I do is I assemble them with a program to assemble transcriptomes, like Trinity, and then I blast them with the NCBI database and check and see what is there.
Okay, thanks.
Yeah, that is doable. Of course, let's put this thing. You have 30% of reads that doesn't map. You assemble them, and that usually take 1 day, more or less. No, it's not more than that. And imagine that after that you assemble them, you have something like, I don't know, 50,000. Of course, to map 50,000 is a lot against NCBI. So the thing that I do usually is I take randomly 20 or 30, and I map this randomly 20 or 30. And if I have a big contamination of something, it's going to tell me, oh yeah, you are mapping human, or you are mapping, or 10% or 20% of these ones that you are mapping are E. coli. Is how you see that these things.
Okay, thanks very much.
Okay, any other question? Okay, so the purpose of sorting BAM files with some tools is to do the next tools more efficient. So the thing is, when you are trying to identify genes, for example, if you have random, so let me put this example here. Okay. So when you see this structure, of course, once you have the mapping, the problem is that you don't have them sorted. Okay. So maybe you have your FASTQ file, and when you map them, you may have these as the first read, this as the second, another of these at the third, another of these at the fourth, another of these of the 5, and so on. So that will be, you have your text, you have that order, okay? The problem is that when you are trying to do anything after, the file, these programs are going to read the file, this BAM file from the beginning to the end. So if when it's reading the file, it's going to go one read, then another, then another, then another. It's going to start to jump from one place to another. It's going to be more complicated for the program. It's going to need a lot of resources just to, for example, count how many reads do I have with the gene. Okay, let's put this example. Imagine that I have this read, these reads, okay? And then I'm counting how many of them do I have in gene 1, gene 2, and gene 3. So I read the first line. I have one for gene 1. Then I read the second line. I have one for gene 2. I read another line. I have another for gene 2. I read another line. And now I have one for gene 3. So you are starting to have these jumps, and that is not efficient. So it's more easy just to sort by coordinate and say, okay, I'm reading for coordinate. For the gene 1, I have 1, 2, 3, 4, 5, 6, 7. Let's move to the next coordinate. For the next coordinate, I have 1, 2, 3. Let's move to the next coordinate. For the next coordinate, I have 1, 2, 3, 4, 5. So it's more easy that start to jump from one place to another, trying to quantify the rates. So it's the reason why you need to sort the BAM files. Okay, perfect. Any other questions? Yeah, if you don't have questions, or well, you can still ask questions. Before I stop the recording, I'm going to talk about the exercise. So let me go to practice. Okay. So for the practice, as I said, I'm going to present the practice here in this recording, and then I will do a different recording for the solutions. So the practice is going to be simple, okay? You are going to do read mapping. You don't have to do it with all the reads, because this can take a lot of time. So you feel that you want to do it, it's fine. You want to do just with some pairs, it's fine too, and I will give you the solutions, okay? So you have, I'm going to propose to use 2 different mappers, and then you can compare both, okay? So you're going to use I2TA and start. And of course, for the following steps, you can use any of those and still for the next steps, and that will be fine. So I2TA is based in this paper. So as I said, this is the Tuxedo, it's called the Tuxedo Pipeline. So use I2TA for the mapping, Slim time for the quantification, and Bargo for the differential expression. And mostly the thing that you have is this process in which you map the reads, and then you modelize the transcripts. You do this assembly of the transcript. I will tell that in the next class. You merge them, and then you compare, and then you will have these coverage tables, and then quantifications, and then you put them in Bargo, and then you calculate differential expression. Okay, so that is the pipeline. So the first exercise is going to be, of course, generate the index. Before you starting to map the reads, you need to generate an index. If you do, perfect. Okay, so you need to do just one. That will be I2TA build, reference, and then the index name that you want to use. I usually use the same than the reference, but I remove the dot faster. Once you have that, you need to map the reads. Okay, and the thing is, by default, this gives you a sum file. The sum files are very big. So the thing that I usually like to do is I pipe the output of this tool into the new command that will be sumtools. So you need as an input the I2TA index of your genome, and then the reads to map in the FASTQ format. And the recommended command will be something like this: I2TA, the number of threads. Please, for the number of threads, I put here 100 because it's the one that I use, but don't use more than 8, okay? Because there are many of you, and it will take, I mean, it will overload the server very fast if all of you use more than 8. DTA, because it's one of the necessary outputs for I2TA. then the reference, x dash x the reference, dash 1 the first pair, dash 2 the second pair, summary file, sum to view, and so on. Keep in mind that in this case, you are going to produce mapped reads to do the assembly, but not to look for the expression. Okay, for look for the expression, then at some point you will need to run a string type with dash E and B. Okay, I will talk about things later. So, and then of course, once you have done that, you need to sort the BAM files. Then once you have that, you are going to evaluate the HiSat results, for example, with BAM toolstats. As I said, that will take some time. The second option is start, and I like start. So one common pipeline that a lot of people use is start HT_SeqCount to quantify them, and then dissect2 for differential expressions. So mostly you have the reads, you use the reference, you need to index the reference first, and then you run start. Once you have the mapped reads, you count them with HT_SeqCount. And then once you have counted them, you calculate differential expression with the sec2. So first you need to generate the index. So here are some commands. Because start can use a GFF file with the annotation, but the GFF file is not GFF, but GTF. First you need to transfer the GFF file in GTF with this command. I usually recommend also to create a directory where you want to put the different indexes of start. And then you run start in this run mode genome generate to create the indexes. It's important to use genome SA index basis 12. By the way, this is an error. This should be dash dash. Okay, so all of these arguments are dash dash. And once you have the index, you can start to map the reads using this. One of the advantages of start is that when you are mapping, the output can be in BAM format directly, and also it can be sorted by the coordinate. So that helps you a lot. And then can be quantified. And then you can go to evaluate the results with start, okay? So that is for the presentation. For the exercise is 11:30, well, past 11:30. So I'm going to give you 1 hour and then some time for lunch. So we can go back with the results and try to resolve the exercise if you want at 1 or 1:30, whatever you prefer. So yeah, let's suppose that we go back at 1, okay? And then if you need more time, just let me know. So 1, you will have 1 hour and a half to start to run this exercise and have lunch. So let me stop first recording before it's too big. Okay.